In this course, I would like to present some of our recent findings on the role of HDL as a pharmacological target in cardiovascular disease and beyond. According to the World Health Organization, atherosclerosis and coronary heart disease are constantly growing problems. Even though in 1996, 7.2 million people died of coronary heart disease, in 2010 this number increased in 9.37 million people, and in 2030 it is expected to rise further to 23.6 million people, suggesting that coronary heart disease is the most common health problem worldwide with an increased prevalence. And this is quite ironic if we consider all medicines that are currently available in the market for the treatment of dyslipidemia. Indeed, dyslipidemia has been identified as a leading cause of coronary heart disease. In particular, an imbalance between atheroprotective and atherogenic lipoproteins have been implicated in the development of this condition. Thus, very wisely, lowering of LDL cholesterol has been used as a main strategy for therapy for the last 30 years or so. Indeed, statins stand as a bulwark in the frontier of therapeutic strategies, being very effective drugs in reducing total and LDL cholesterol levels. However, recently it became apparent that statins are associated with residual risk. Residual risk refers to the fact that even though one may enjoy a very significant reduction in total and LDL cholesterol level, this person is still under significant risk for developing a fatal or non-fatal myocardial infarction. Even though different studies place residual risk at different levels, it appears that on average, approximately 70% of people who take statins will develop a myocardial infarction event at some point in their lives. Therefore, it appears that even though statins have given great results thus far, their benefits in terms of reducing coronary heart disease mortality has reached a plateau. So we need new medicines. We need new medicines that ideally will efficiently decrease coronary heart disease related mortality to all treated patients compared to the current medications, and prevent plaque formation, stabilize unstable atherosclerotic plaques, or even cause regression of established lesions. However, before we go ahead and design such pharmaceuticals, it's very important that we decide on the pharmacological targets. In the effort of identifying new pharmacological targets, HDL has been identified as an important lipoprotein in the prevention and treatment of coronary heart disease. And this is based on the classical view from the early studies of John Goffman and Richard Havel that were further confirmed later by the Framingham Heart Study, showing that if someone has elevated HDL cholesterol levels, that is 40 milligrams per deciliter for men and 50 milligrams per deciliter for women, then this person is protected from developing coronary heart disease. Therefore, based on this principle, a number of experimental pharmaceuticals are currently under development, such as CDP inhibitors, ApoE1 Milano, ApoE1 mimetic peptides, inducers ApoE1 expression, EL inhibitors, and delipidated HDL. Most of these experimental drugs focus on raising HDL cholesterol levels. But the question is this the right thing to do? In other words, is HDL cholesterol the appropriate pharmacological target? Looking at the recent studies on torcetrapib and dalcetrapib, where an increase in HDL cholesterol levels did not correlate with improved atheroprotection, maybe raising HDL cholesterol levels is not the right thing to do. However, we still have to wait for the outcomes of the REVEAL and ACCELERATE trials in order to decide whether raising HDL cholesterol was a successful idea or simply a failed attempt. In any event, a recent meta-analysis study published in The Lancet showed that SNPs associated with increased HDL cholesterol levels may not be used as surrogate markers of atheroprotection. In other words, SNPs on HDL proteins that correlate with increased HDL may not be markers of improved anti-atherogenic potential of HDL. Based on all these findings, recently the principle of HDL quality was proposed. That is, in addition to HDL cholesterol level, HDL quality and functionality is also very important for atheroprotection. So, according to this principle, HDL is composed of proteins and lipids which all contribute certain properties to the HDL particles. Therefore, HDL sits in a way on a balance, and depending on the properties that these HDL may have, the balance may tilt towards atheroprotection or no atheroprotection, or HDL may even be pro-atherogenic under certain conditions. So, in an effort to identify potential HDL targets for HDL pharmaceuticals, we decided to ask ourselves, what do we really know about HDL? Certainly, HDL is not just a cholesterol. Cholesterol is one of the lipid components of HDL, which is a particle composed of both apolipoproteins, triglycerides, cholesterol, cholesterol esters, and phospholipids. And this particle may be of different shapes and sizes. So it may be small HDL, it may be large HDL, it may be spherical HDL, or it may be discoidal HDL. In addition, it is very well established in the literature that the main apolipoprotein of HDL is ApoA1. ApoA1 is synthesized primarily by the liver, secreted into the circulation, and travels through all tissues where on the surface of cells it interacts functionally with ABCA1, a lipid transporter which upon dimerization transfers phospholipids and cholesterol onto ApoA1, resulting in minimally lipidated ApoA1. Then, through a series of steps that are not very well understood yet, this minimally lipidated HDL is converted into discoidal HDL, which with the action of plasma enzyme LCAT is then converted into spherical HDL that possesses a number of anti-atherogenic properties. And also, this spherical HDL can functionally interact with SRB1 and unload the HDL cholesterol that it carries. However, analysis of HDL preparations from plasma have shown that in addition to ApoA1, and approximately 70 other proteins are also present on these HDL preparations. So the question is, are all these proteins present on a single particle, or different particles carry different proteins? Therefore, to address this question, we ask if other apolipoproteins different from ApoA1 are also capable of promoting the de novo biogenesis of HDL particles. And as an example, we used apolipoprotein E. In this set of experiments, we did gene transfer of ApoE in ApoE1 deficient mice and studied whether expression of ApoE can promote the formation of HDL. Indeed, mice received either wild-type ApoE4 or a mixture of ApoE4 and LCAT using adenovirus-mediated gene transfer. The next slide is a little busy, but I would like to focus your attention primarily on panels H and I. Panel H shows that overexpression of ApoE in ApoE1 deficient mice results in very nicely shaped discoidal particles, and then panel I shows that co-expression of ApoE with LCAT results in the conversion of these discoidal particles into nice spherical HDL particles. And this is a process that heavily depends on ABCA1, because in the absence of ABCA1, ApoE4 is not anymore capable of promoting the biogenesis of HDL-like particles. So based on this experiment, we concluded that ApoE can functionally interact with ABCA1, promoting the formation of initially discoidal and later spherical HDL-like particles. And now we know that in addition to ApoE, ApoC3 is also capable of promoting HDL-like particles. From this analysis, we concluded that HDL is a mixture of particles with distinct apoprotein composition and densities in the HDL region. In other words, we concluded that different particles may carry different apolipoproteins, and that the composition of each one of these particles may differ from the composition of the other particles. So next we wanted to know what do these distinct HDL subpopulations may mean for human physiology and disease development. For this reason, one example that we looked at was biliopancreatic diversion by Roux-en-Y with long limbs. This is a restrictive and malabsorptive bariatric procedure usually used for the treatment of morbidly obese patients. The nice thing about this procedure is that it associates with significant reduction in coronary heart disease risk 10 years following the operation. Indeed, what we found is that 6 months after the operation, we had a significant increase in the antioxidant potential of HDL that correlated with a significant reduction in CETP activity. And as you know, reduced CETP activity correlates with atheroprotection, and this is why the biomedical community focused on inhibiting CETP as a potential way of treating coronary heart disease. When we performed some analysis of the apolipoproteins present of the HDL particles of these patients, we were very surprised to find out that in 35% of the patient cohort, HDL was mainly present in ApoE containing particles. Very little ApoE1 and almost no ApoC3 was present in these particles. 6 months after the operation, we had mainly ApoE1 containing HDL particles with very little ApoC3 and ApoE present on these particles. Now, in the remaining 65% of the patients, we found that HDL cholesterol was mainly present in ApoE1, ApoC3, and ApoE containing particles. However, again, 6 months after the operation, HDL cholesterol was mainly present in ApoE1 containing HDL particles. And just to give you an idea of what the structure of the composition rather of the HDL in apolipoproteins in healthy individual is, in panel A of this slide, you see that healthy individuals have primarily ApoE1 containing HDL with trace amounts of ApoC3 and ApoE present. And this is the exact picture that we got from the obese patients after the operation. When we quantify the relative apolipoprotein ratios, we saw that following the operation in both 35 and 65 cohorts, there was a significant increase in the ApoE1 over ApoC3 and ApoE1 over ApoE lipoprotein ratios. So from this type of analysis, we concluded that having 2 too much ApoE or ApoC3 in your HDL may not be good for the function of this HDL. The other example that we looked at was young asymptomatic patients who suffered an acute myocardial infarction. In other words, here we performed structural analysis of HDL in a group of patients who were younger than 35 years of age. They had no previously diagnosed coronary heart disease. They had no symptoms and no comorbidities that could contribute to the development of the disease. Analysis of the antioxidant potential of HDL of these patients showed that rather their HDL was pro-oxidant, so their HDL was promoting oxidation and had no antioxidant capacity. Also, we saw that the relative ApoE1 over ApoC3 and ApoE1 over ApoE ratios of HDL in these patients were significantly reduced. And I would like to remind you that in the obese patients following the operation, we saw the exact opposite thing. Again, confirming that having too much ApoC3 or ApoE in your HDL may not be very good for the functionality of your HDL. So, based on these findings, we propose that HDL apolipoprotein ratios correlate with HDL subpopulation distribution and functionality. And most importantly, we propose that apolipoprotein ratios may be a useful diagnostic tool for determining HDL functionality. We are very curious to know what pharmacological modulation of such ratios may mean in terms of HDL functionality, and so we are currently exploring this possibility, and in order to validate the role of such ratios in HDL functionality. In addition, we believe that in the evaluation of any HDL-based pharmaceutical, it is very important to determine also the relative apolipoprotein ratios and the effects that this pharmaceutical has on these ratios in order to define efficacy of such pharmaceuticals on HDL functionality. And here I would like to thank all our friends and collaborators who contributed to this work, and also acknowledge funding from the different funding bodies who supported our research all these years. Thank you very much.